Okay, we're going to be talking about oxidative phosphorylation. So right here we have a eukaryotic cell. Notice it has a nucleus and a mitochondria. Now if you look at the mitochondria, you notice that it has these little bumps in the center of it. Let's take a closer look at one of these and let's see what we find. Well, in here we have the outer mitochondrial membrane right here, and then the inside part, this wavy part, is the inner mitochondrial membrane. Now these bumps, or waves, increase the surface area of the inner mitochondrial membrane, and we call those waves cristae. Now let's take a closer look at these little dots on the inner mitochondrial membrane and see what they are. So here we have the inner mitochondrial membrane right here, and it's performing a complex task called oxidative phosphorylation, or phosphorylation. It's divided into two parts. The electron transport, which is characterized by this long chain of dotted lines, and then chemiosmosis, which is characterized by this organelle right here. Now let's just walk through the process real fast. We have NADH present within the mitochondria, and it has two extra electrons on it, and it moves through these proteins here on the inner mitochondrial membrane, which can then take those electrons from NADH, which renders NADH to become NAD+. Now, these two electrons move through the dotted lines here, and as they do so, they release a little bit of energy every time, and that energy allows for one of these hydrogen ions, or protons, to move through the protein there. Now, it is worth noting that that the electrons don't enter only through the protein labeled 1, it can also enter through the protein labeled 2 with FADH2, in which case the FADH2 has two electrons, it drops off the electrons and moves through the chain, and FADH2 becomes FAD. By the way, we get electrons in the electron transport chain. And as it moves through the chain, it keeps moving along. Each time it passes one of these proteins that pass through the membrane, which are proteins 1, 3, and 4, it releases enough energy to move a hydrogen ion across the inner mitochondrial membrane. And eventually it passes through 4, which releases another hydrogen ion. And now we have these electrons that release a lot of energy and don't have much left in them. In which case, we have an oxygen oxygen molecule here that come and pick up the electrons that have just exited the chain into the inside of the mitochondria. And since this oxygen atom has a negative charge, it attracts two more hydrogen ions into it, which form water, H2O. Now, I want you to notice how the proton gradient now that exists. We have a lot of proteins on this side of the inner mitochondrial membrane, but very few on this side. This creates a proton gradient where the protons want to move from where it's more concentrated to where it's less concentrated. And this is a term called chemiosmosis, where the protons act in an osmotic way, where it moves from more dense to less dense. And the protein, or series of proteins that allow that to happen, is called ATP synthase, which is right here. What happens is the protons, or the hydrogen atoms, move down its concentration gradient, and enter into this little tube right here, which ends up spinning as it moves in and then eventually gets moved into here. Now that spinning creates energy as it happens, and then when that energy is released, ADP and non-organic potassium enter NTP synthase. That energy allows it to become ATP, which is the basic energy unit of cells, which is then released. And that describes oxidative phosphorylation, or moving the phosphate to ATP with the help of, that involves oxygen. Now, this is only a small look at cellular respiration, and there are several items that we might be questioning why they aren't there, such as where are glucose, carbon dioxide, pyruvate, and acetyl-CoA. While these components and molecules are very important to cellular respiration, they occur earlier in the cellular respiration process and do not directly have involvement with oxidative phosphorylation. Thank you.